All right, moving on. Our last internal speaker is Dr. David Wald. David is a highly accomplished physician scientist with a research focus in translational cancer research, in particular with an emphasis in hematopoietic cancers. He is a professor in pathology and scientific director for Wesley Center for Immunotherapy. He's also a co-director of our institutional hematopoietic biorepository and clinical cell therapy lab. His current work is heavily focused in the development of new cellular therapy products and biomarkers for heme malignancies. His research have been very high impact, published in a number of high impact journals, numerous NIH and VA-funded proposals, a number of prestigious awards, and a plethora of patents under his belt. Today, he is going to talk to us about strategies to improve CAR T cell therapy. David.
Thank you very much for the introduction and for the opportunity to talk about my work today. So I'm going to talk to you about two different strategies that we're taking in the lab to try to improve CAR T therapy. And just as a disclosure, I have an involvement with a small startup company that does some CAR T cell therapy work. So I'd just like to start out by giving an introduction to what is CAR T therapy, or chimeric antigen receptor T cell therapy. And so this is a genetic engineering of T cells, and you can see a schematic here of what a CAR, a chimeric antigen receptor, looks like. And so this consists of an extracellular domain. This is the antigen binding domain. Typically in the classic CAR, it involves an antibody sequence called a single chain variable fragment derived from antibodies. So this is what binds to the antigen you're trying to target. For example, in our case, a tumor cell antigen. This antigen binding domain is then connected to a hinge domain that gives flexibility to allow this sequence to actually target the antigen of interest, and then a transmembrane domain, so it inserts in the T cell membrane. And then you have a series of co-stimulatory domains. And so we're utilizing primarily for our work what's called a second generation CAR, which involves two signaling domains, classically a CD3 zeta domain, and then a co-stimulatory domain such as 4-1BB. And so in this way, when you engineer a T cell, it's now able to recognize a antigen that it's normally not able to recognize. So in the case that we're going to focus on today, it's the CD19 antigen found on B cell malignancies. And so these T cells now can interact with these tumor cells, and because of the signaling domains and the intracellular domains, it's now able to actually target these cells for lysis and kill the target cells. And so the engineering of this therapy starts typically with collection of blood from a patient. Currently, the FDA-approved CAR T therapies are all autologous therapies, and so you start with the patient, and typically you perform an apheresis or leukapheresis procedure to obtain large numbers of white blood cells and a source of T cells to start the cell therapy engineering. And then you take them to the lab, and we're fortunate here at our institution to have a really great cell therapy lab, actually on the 6th floor of this building. We are about to open a major expansion of the lab in the next few months, where we can perform clinical grade manufacturing of the product to keep the product sterile. And in most cases, and in all the cases the FDA approved products, the engineering of the T cells involves a viral mediated gene delivery into the T cells. And so people use typically either lentivirus or retrovirus to insert the CAR gene into T cells. And so now you've created your CAR T product. However, under classic manufacturing processes, the cells are then expanded, usually between 1 and 2 weeks, to generate large numbers of cells for patient infusion. Okay, and then the final step, obviously, is then to reinfuse the product, either a frozen product or a fresh product, back into the cancer patient, where it's hoped these T cells will actually migrate to the tumor and have significant anti-cancer activity. Okay, so as I mentioned, I'm going to focus today talking about our work on CD19 CAR T therapy. And I believe this has really been one of the remarkable areas in the recent history that have really revolutionized cancer treatment and outcomes for patients. And CD19, as I mentioned, is expressed on B-cell malignancies, so that includes lymphomas as well as leukemias. I'm going to focus primarily talking about its role in non-Hodgkin's lymphoma. So non-Hodgkin's lymphoma is the most common type of blood cancer. There's over 500,000 patients diagnosed per year worldwide. Unfortunately, about 30% of patients still die within 5 years of diagnosis. And this is primarily due to the fact that around 40% of these patients will develop refractory or relapsed disease. And it's particularly in these patients, as it is true in most cancers, where patients who have relapse or refractory to initial chemotherapy have very poor responses and outcomes. So in this population with traditional chemotherapy, it's been reported that there's a 7% overall response rate to chemotherapy and about a 6-month median overall survival for the most common subtype of non-Hodgkin's lymphoma, which is called diffuse large B-cell lymphoma, or DLBCL. And it's in this particular population of patients that have shown the relapse refractory status to chemotherapy that CD19 CAR T cells have shown remarkable efficacy. And so in this population, people have reported over 50% remission rates. We, as I'll talk about, a clinical trial conducted at our cancer center actually saw, I think it was around 80% remission in this population. And so there's been really remarkable outcomes and made great improvement, I think, to lives of many patients. However, that being said, there's still major problems. So even though I said about greater than 50% of these patients are attaining remission, these remissions are not durable long-term for most of the patients. So after a year, I think it's about 50% of these patients will unfortunately relapse. And this slide is trying to highlight some of the key challenges that we still have in CAR T therapy despite this initial success. And so you can see on the left side of the slide, the first major challenge is unmet needs. And so CAR T therapy has been very effective for B-cell malignancies and also multiple myeloma. There's been successes, but for most other malignancies, we've yet to see similar levels of clinical efficacy. And so, increasing the success of CAR T therapy for other malignancies like brain tumors or other solid tumors is a major challenge in the field. Another major challenge is toxicities. And so even though these are successful treatments, they're not without significant and in some cases life-threatening toxicities. These include neurotoxicities. This is thought to partially be a result of the fact that CD19 can actually be expressed in some brain tissue. And you can also get life-threatening cytokine release syndromes. And so developing modified approaches to lessen these side effects is very important. And the clinicians have gotten much better at managing these toxicities, but they still do occur. Another major challenge, as I alluded to earlier, is late relapse. And so though there's a very high initial remission rate, there is late relapse. And in cases of leukemias, acute lymphoid leukemias, this is often due to antigen loss. And so when the tumor cells initially express CD19 and then you infuse CAR T cells into the patient, they're effectively able to lyse cells, but the tumor cells are smart and they evolve and they downregulate the surface expression of CD19 and then develop resistance. And then finally, what I'm going to focus on today is actually failure of the CAR T, intrinsic failure of the CAR T cell product itself. And so when we infuse CAR T cells into patients, unfortunately, in many cases, they do not persist long-term. And this persistence of the CAR T cells in vivo in patients is really correlated with long-term efficacies. Okay, and this is highlighted by a study that was reported by Dr. Michael Green a couple of years ago, where he looked at properties of the CAR T cell product that was infused into patients. And the bottom line was that the product itself really had a strong correlation with outcomes. And so if you started with a product that had a more naive memory phenotype of the T cells, these cells were able to persist much longer in patients, and this correlated with efficacy. So you can see in the diagram here, in blue, we're looking at the patients who had complete remissions, in red, partial remissions or progressive disease. And the complete remissions are dominated by T cell products that are enriched for memory CD8 cells and memory CD4 cells. And the patients who had worse outcomes are characterized by T cells that were exhausted. Okay, and so as I mentioned, we had completed a clinical trial. This was run by Paulo Cami and Marcos de Lima at the Seidman Cancer Center with a CD19 CAR-T product that was manufactured using the Miltenyi Prodigy, so it's an automated manufacturing platform. And they treated 30 patients with non-Hodgkin's lymphoma in this phase 1 trial using a CD19 CAR, a molecule similar to the one I showed at the beginning of the presentation. And what we did is we collected samples from these patients, either the manufactured CAR-T product or blood samples from the patients at a variety of time points after infusion of the product. And the goal was to understand what changes these CAR-T cells underwent in vivo, and can we then learn from these changes to predict how patients will respond and then hopefully eventually develop better next-generation CAR-T therapies. And so in this schematic here, I'm trying to illustrate some of the analysis we performed on these samples. And so the first step was just to isolate the CAR-T cells, and so we performed flow cytometric sorting so we could isolate the CAR-T cells both from the product itself as well as from the blood samples from the patients. And in this particular study, we looked at day 14 and day 30 patient samples after infusion. And then we performed a variety of different multi-omics type studies on these cells after we collected them. And this include single-cell RNA sequencing, as well as protein assessments by a process called feature barcoding. And then we did a variety of different analysis, which I'll show you a little bit of today, to try to assess the differential expression and gene set enrichment analysis to understand what pathways are being modulated in these CAR-T cells after infusion. We also did independent validation studies using flow cytometry to look at protein expression and a separate aliquot of samples. Okay, so I'm just going to show you a few highlights from this study. And so one of the first observations that we made is that after infusion of the CAR-T cells, as shown by many other groups, the CD8 CAR-T cells proliferate markedly after they see the tumor antigen, and they also become exhausted. And so I'm showing you on the left side of the slide RNA sequencing analysis, and on the right side flow cytometry, which are both showing you that we get upregulation of these checkpoint molecules such as PD-1, CTLA-4, TOX, and TIGIT that are known to downregulate T cell activity. So you can see, both by RNA sequencing and protein expression by flow cytometry, we're seeing the same results. And in particular, PD-1 and TIGIT were the most upregulated checkpoint molecules observed after infusion of the CAR-T product. However, we wanted to understand why is this product working much better in some patients than other patients. And so we segregated our patients into favorable responders and poor responders. And favorable responders were patients who underwent a remission, either a partial remission or complete remission, for purposes of this study. And interestingly, what we observed is when you actually look at outcomes, there was no difference in PD-1 expression across the outcome, poor responder or favorable responder groups. However, there was a significant increase in TIGIT in the poor responder groups as compared to the favorable responder groups, both at day 14 and day 30. And we observed that by both RNA sequencing and flow cytometry analysis. So we did a variety of other studies to show that these cells that have upregulated TIGIT are actually functionally exhausted T cells. And then we hypothesized that we may be able to take advantage of this upregulation of TIGIT for therapeutic purposes. And so could we actually block TIGIT or abrogate TIGIT and improve CAR-T therapy? And so we did a variety of in vitro studies, which I'm not going to show you today, as well as a number of different mouse models. And this is a representative mouse study showing that we can abrogate TIGIT using a therapeutic monoclonal and improve CAR-T efficacy in a mouse model. And so this particular model is a mouse model of human lymphoma. So we take a human lymphoma cell line called Raji. It's engineered to express a marker called luciferase so we could track the disease burden in the mice. The mice are injected intravenously with the tumor cells, so the tumor becomes systemic. And then a week after tumor injection, we inject CAR-T cells and look at disease burden as well as survival. And what we found is that we inject CAR-T cells, and this is a high tumor burden model, so we purposely use a mouse model where actually, in this case, we waited 3 weeks after tumor cells were injected, so we had a very high tumor burden model, and CAR-T cells normally are not very effective. And you can see in the graph here, there was no difference in survival with the CAR-T cells in this particular setting. However, when we block TIGIT with an antibody from Bristol Myers that's being currently used in clinical trials, there was a significant increase in survival. Okay, so I'm going to change gears a little bit now based on that study. So that initial study, I think, is an example of some of the work that we can do at our Cybin Cancer Center, where we are able to conduct clinical trials and then take that patient material back to the lab to hopefully develop new therapeutic strategies that we hope one day in the next few years we could then lead to another follow-up clinical trial where we combine our CAR-T product with a TIGIT blockade strategy. And so we're currently working on those approaches in the lab now. Another major hurdle, besides the exhaustion of T cells, as I mentioned before, is the persistence and the continued presence of the T cells in the recipient at long time points, for example, a year after infusion. And as I mentioned earlier, the study by Dr. Michael Green, which showed that this is really correlated with the memory status of the T cells. And so what we've done is we've recently developed a rapid manufacturing process. So I mentioned previously the typical T cell manufacturing process is about a week or two of culture in the cell therapy lab. And during that culture period, the T cells actually differentiate. You lose a lot of the naive cells and the central memory T cells that are really critical for this long-term persistence. And so we've now modified that protocol to have a 20-hour manufacturing process. So in a single day, we're able to generate a CAR-T product that then can go into the patient. And by doing this, we can have a much higher level of the naive T cell population that had differentiated during the culture process. We think there's a number of advantages of the rapid CAR-T manufacturing process. In addition to maintaining the memory T cells, we hope that this may also address some of the side effect profiles that we see with cytokine release syndrome. So because we're now infusing an immature product into the recipient, these cells are not actually mature. They're not as functional at killing the tumor cells. They have to go a process of in vivo maturation. And so the kinetics of tumor killing is going to be reduced, and we hope that that will correlate to a reduction in cytokine release syndrome that really correlates with a very rapid tumor kill. We also think that this process can also allow this therapy to be much more accessible to patients. Currently, it can take 3 to 6 weeks, or in some cases months, for a patient to have CAR-T cells manufactured. And so by doing it in a single day, we can get the product to patients quicker, which is very important for patients with relapsed refractory cancer, where outcomes can be dictated by how quickly they get treated. The current manufacturing processes are also very expensive. It costs about $400,000 in the United States for this product. Worldwide, this makes this very inaccessible to most patients worldwide are not able to access this life-saving therapy. And so by making it more simple and inexpensive, we hope it will become a lot more accessible. And I'm just going to show you kind of a single data slide from the rapid product that we created. This is a mouse study just showing, sorry, some of the graph, it seems like it didn't format correctly, but showing that the survival of the mice is actually significantly improved using our rapid CAR-T product as compared to a traditional manufactured product. And so in mouse models, when we compare a similar dose of the rapid product versus, in this case, a 6-day manufactured CAR-T product, we see significantly less tumor burden and significantly better survival with the rapid product. And this also correlates to the number of T cells we see circulating in the mouse model. So we see 10 to 100-fold more T cells persisting in the mice as compared to the standard products. And so we've recently initiated a clinical trial that's being run by Dr. James Martin here. We hope to accrue our first patient in the near future, but we hope that this will also translate into the human setting as well. Thank you very much.
You have time for 2 questions. Grace?
Hi, very nice work, actually, even for an ID person. Does it work in CNS lymphoma? And then if somebody had relapse, can you do it again with or without TIGIT antibody?
So CNS, I haven't worked personally on CNS lymphoma. Probably Dr. Van Biesen would probably better address that than me, since I'm a pathologist, not an oncologist. I don't see those patients. But in terms of relapse, that's a good question too. I think it depends on why they relapsed. If they've lost CD19 antigen, then it's not going to work to infuse another product. In this particular case, we are excluding patients who received a previous CD19 CAR-T therapy and relapse, because those are very difficult patients to treat, and it'd be hard to interpret, I think, the data in our small phase 1 study. But there's a lot of strategies out there where people are going with bispecific CARs directed against two different antigens or other antigens besides CD19 to address those issues when people do relapse. There's alternative approaches that can be taken.
I was going to ask a question, but I'll answer the question about CNS lymphoma. There's some rumor out there that it works, and we've treated patients. Most of patients with CNS lymphoma were excluded from the original trials, but some kind of made it in, and there's responses. How good they are and how probably a degree less, and that's been my experience. Probably more prone to these neurotoxicities that you mentioned. David, I also have a question about, you mentioned cheaper and more amenable, but that's indeed a big question, particularly in resource-constrained countries. Is this a closed system? Is this the Miltenyi device that you use again, or?
Sure. So it is not using a Miltenyi device, and we specifically designed this workflow to avoid any expensive equipment so that we could keep the costs very, very low. So they're probably about 50 times lower than the current commercial processes is what we're estimating. We're in the trial that we're doing now, it's not a fully closed system, but we're currently working on making it into a fully closed system. I think it's pretty amenable to make it fully closed. The long-term goal would be to be able to have a process that we can actually do outside of a cell therapy lab and even a community hospital, or to make it. Even in the United States, there's accessibility issues when you go outside of the major academic centers.